Welcome to this third lecture. In this portion, we will discuss about this endomembrane system. I hope you have heard about this endomembrane system in class 9th itself. Now we will go little further. Okay. So what is endomembrane system? You can see this as endo and membrane. Two words, right? Endo means within. Okay. Within what? Within cell. Within cell, there are many membranous structures that are interconnected with each other, and these structures are known as endomembrane system. They perform a particular function. As you can see, there are organized function performing by all this membrane system. Here is the definition if you want it. Then the endomembrane system is a group of membrane and organelles in eukaryotic cell that work together to modify, package and transport the lipids and proteins. I hope you must have heard about this term lipid and proteins in digestion chapter in your 10th standard. So what is this modification, packaging and transport of this protein? For example, you can say if our cell needs to secrete certain enzymes, or it as you have already know that some of the glands secrete the hormones, right? So hormones are sometimes protein also. So cells are secreting this particular protein. So sometimes what happens, where synthesis does occur of the proteins, you know this as rough endoplasmic reticulum or the ribosome part, right? The rough endoplasmic reticulum, where in the endoplasmic reticulum the ribosome is present who synthesizes the particular proteins. After the synthesis of protein, what happens? Sometimes we need to modify that particular protein. Like we can attach some of the molecules in that, we can detach some of the molecule in that. We will discuss in further lectures. And after that, we package that particular protein where we want to send it. Okay. And this particular transport of lipids and protein. I have given an example of protein. You can take the example of lipids also. So let's see what are the particular organelles that are involved in this particular endomembrane system. The organelles are, first of all, the endoplasmic reticulum. You know that endoplasmic reticulum are of two types. First one is the rough endoplasmic reticulum, or in short you can say it as RER. And second one is the smooth endoplasmic reticulum, or you can say it as SER. The second cell organelle is the Golgi apparatus. Third one is the lysosome, and the fourth one is the vacuole. You know all this cell organelle, right? So let's take this particular example of how a protein is synthesized in the cell, and after that how it is packaged and modified, or it is secreted out of the cell. Here you can see the nucleus part. We are talking about eukaryotic cell. Just mind it, okay? So this is the nucleus part. I have drawn the DNA or chromatin material, whatever you can say. This is the outer nuclear membrane. As you must have known that nucleus is a double membrane structure. This one is the outermost membrane and this one is the innermost membrane. And this is a rough endoplasmic reticulum. Remember this one, that the rough endoplasmic reticulum always continues with the outermost membrane of nucleus, okay? Here you can see this whole structure is the rough endoplasmic reticulum. In the rough endoplasmic reticulum, you already know that ribosomes are present on the surface. So what ribosome does? Ribosome synthesizes the particular protein molecule. After synthesis, what happens actually? It goes inside this vesicular structure of this endoplasmic reticulum, and from this endoplasmic reticulum, small vesicle. This you can see what is mean by vesicle. These are the balloon-like structures, like this membrane-bound balloon-like structures. Inside this, whatever protein has already been synthesized, this will be packaged. Okay, like package you must have seen in the food delivery processes. Restaurant do that. Okay, like that. After this formation of vesicle, what these rough endoplasmic reticulum do? It will send it to the Golgi apparatus. Okay. Now, this is a very important step. Golgi apparatus is also known as the postman of the cell. What does postman do, or post office actually? These are known as post office of the cell. What does post office do actually? Post office will tag the address of destination wherever you want to send a particular letter, right? Then it will send it like that. After this vesicle, when this vesicle will fuse with this membrane like this, and after that what happens, these vesicle pass from this, and then inside this process the Golgi apparatus attaches some of the molecules like this on the vesicle. So what happens after that? These molecules contain the information where it will go. If there is a information of going outside of the cell, then after coming out of this Golgi apparatus, it will go out of the cell. If it has the information of going inside the mitochondria, like sometimes the mitochondria needs some enzyme, then what will happen? This particular vesicle will go inside the mitochondria. If wherever this information will lead it will go inside that particular organelle or outside of the cell. Got it? Here I have described how the rough endoplasmic reticulum and the Golgi apparatus are interconnected. Like that, you can see this diagram. This is the plasma membrane. Okay, this is the plasma membrane. This is a foreign molecule, or you can say any food particle, or any foreign microbe you can say. Okay, this is a foreign microbe. When it encounters this particular plasma membrane, it comes inside it. Here you can see the invagination of the plasma membrane, like endocytosis process. You must have learnt about endocytosis process in your 9th standard. So here this particular microorganism has entered this inside this endocytic vesicle. After what happens? This endocytic vesicle was cleaved up here. Okay, it will cut down, and here it has formed this vesicle here like this. Like this vesicle form, and here we the foreign microorganism that has come inside. Okay. Here after this this is the lysosome, and what it does, lysosome will come and fuse with this particular vesicle, endocytic vesicle. So you must have known the lysosome contain all the hydrolytic enzyme, right? Many hydrolytic enzymes, like this has the enzymes that break down the protein, the enzymes that break down the lipids, the enzymes that break down certain carbohydrate molecule, all that. So this lysosome now fuse with this particular endocytic vesicle, and this will form a common structure. Here you can see a red part, right? Red part is from the lysosome, and this blue part is from this endocytic vesicle. After the fusion, here this lysosome has already the hydrolytic enzyme. So now this hydrolytic enzyme will degrade this particular microorganism. Okay, it will degrade the particular microorganism, and after that what it does, it will send it out of the cell. Here I have discussed about in this lecture. You can see here this endomembrane system. Endomembrane system contain 4 cell organelle, that are the endoplasmic reticulum, Golgi apparatus, lysosome and vacuole. These 4 cell organelle function simultaneously or interrelatedly. Okay, here I have discussed about how this particular endoplasmic reticulum and Golgi apparatus are doing a great job, and what happens, sometimes the lysosomes are formed from like this. Okay, so these 4 structure are interconnected. Like vacuole, what vacuole does, as you already know, vacuole maintain the osmotic pressure of the particular cell or plant cell. These vacuoles contain certain cell sap, that are the sucrose molecule, that are certain ion channels, that are certain ion molecules like sodium ion, chloride ion, potassium ion, all that are present inside the vacuole. And what it does actually, it helps in the building of the osmotic pressure in the particular cell.